Good afternoon and welcome to Transcell's second event in what we plan as a series of events. Um, we'd like to welcome the participants, primarily our guest, Dr. Subramaniam Vangala. Dr. Subramanyam Vangala is CEO and founder of Reagene Biosciences. Uh, and he's an accomplished scientist and an accomplished industry leader. And we'd be happy to pick his brain this afternoon. Dr. Vangala is currently ICMR, is one of the members of the ICMR expert committee for alternatives to animals in research, and a recent and has been the recent contributor to an article on this subject. The article, for those of you who are interested, is titled "The Need for Alternatives to Animals in Experimentation: The Indian Perspective." He's also part of the CSIR team for the review of CSIR-funded drug discovery programs. He's part of the executive committee for the Society for Alternatives to Animals for Experimentation in India. He's also part of multiple other editorial and advisory boards and is very active in the industry. Uh, uh, his current research activities include the development of 3D bioprinted human tissues as in vitro alternatives to animal approaches, uh, with the intention of enabling better translation of discovery to successful clinical outcomes. These models will support drug discovery, nutraceutical, agrochemical, cosmetics, and environmental pollutant research. I'd now like to introduce Dr. Subhadra Dravida, my co-host and founder, CEO of Transcell Oncologics. She manages the various verticals in the company, including Transtox Bio. Transtox Bio is a collection of configured human surrogate in vitro cellular and tissue platforms. Transtox Bio uh, positions itself as a provider of these systems to industry and also as a specialty CRO for the safety and efficacy testing um, of various test candidates. Transtox Bio caters to global pharma, vaccine, and cosmetics industries, and to their in vitro testing requirements. Um, it also promotes, in the process, animal-free or cruelty-free testing methods and models in the industry for the industrial preclinical research and developmental needs. So, with that, I'd like to open the conversation, and I will now yield the floor to Dr. Dravida. Dr. Dravida.
Thank you, Lakshman, and thank you, Mani. To have accepted and giving us time in terms of talking to us, and of course, like whoever is watching this show, like that we tend to telecast as well. So I have few questions, and then in fact, like I've gone about your profile, and then we have followed your trajectory from academia to industry. So, do you think the German model is relevant to India?
Yes or no? The question is that the question comes here is that which model is important is not the question at all for me, and because I worked in hierarchical models as well as in the uh, flat models. The flat models are I like them better. The reason is because you can make your own decisions. Nobody is going to tell you what to do every day and things like that. So European models more hierarchical I've seen, and whereas US models, especially working for J&J for 6 years, it was a very flat model. The between senior VP and me, there was no VPs and other layers, and I had my own ability to make decisions, and I took directly to the scientists and made the work done. So the flat organizations are more productive, uh, more independent, more team working abilities that you have. So I don't like hierarchical models, for example. And if you look at, I think the important thing that we need to look at the shareholders versus the stakeholders, right? And if you look at, I don't know if I can post it here now, but in J&J they had a credo. The credo, you have to follow it, and if you don't follow the credo, then you may even get a poor performance, even if you perform very well. The idea there is that you are only liable to, sorry, you are only accountable to the patients, not to the shareholders.
I
That's
see.
very clear in their credo. And shareholders come last, and the patients come first.
I like it. Yes.
Right, and the employees come second. Okay, so the that's the way they follow it, and every year we had a credo survey, and to assess the entire company. I mean, how is your manager? How is your senior manager? How is your president? What is the system, and how do you like it? Uh, people do people listen to you, and what are your concerns? It's anonymous, and nobody knows who actually gave these surveys, and the entire company does it. It's through a third party collaboration. So the thing is, those kind of checks and balances should be there, so that the communication is not top down, but down up also. So that's the kind of a system I like it, and that's where I liked J&J very well, because that gives you more management depth and how to manage people. I mean, no, everybody is different, and you have to manage each person differently, and you have to change your way of treating them. And some people are easy to work with, some people are very difficult to work with, but it's up to a manager how you work with them, because you can take a whip and beat them, but tomorrow HR is going to come and question you. Why are you treating your person badly? So you spend more time with HR than doing actual work. So the system is so streamlined there, and it was easy to work with each other without too many layers and hierarchy.
Sure. Awesome. Thank you.
Yeah.
So the next question is, where do you think the in vitro models industry is headed?
In my opinion, personally, being in the in vitro area for 40 years now, I mean, through my academic research as well as in the industry, I think it's headed towards more physiological models, which are essential. I'm going to talk about it in more detail, and why that is very essential, because especially I have noticed correlations from in vitro experiments to in vivo, especially for clinical outcomes, has been poor for the last 30 years I've been in the industry. You take in 1990s when I joined in the industry, uh, we were doing.
So the question continued here is, so do you want to blame the in vitro model or the methodology that is kind of developed on those models in assaying? So there are two things here, right? I mean, one is the platform, and the other one is the method of testing. So what is it that, like, probably one should be working at or reworking at or configuring in order to get the extrapolation meaningful to the actual clinical scenario?
I don't blame anybody. The knowledge is area.
Okay.
Okay. Unfortunately, we are still accumulating data, and we don't know how to make any relevance of that to the clinical. Yeah, you've got all this data. What does it mean in terms of the clinical relevance?
Right.
This is where artificial intelligence is now coming in and trying to plug in all this information together with the big data and everything. But still, we are miles away, because the in vitro is the validation that comes in. But anything you validate finally is only in the patient. That's the validation. That's the real validation. If so, my validation in the in vitro or in vivo, in 3D model or even animal, it doesn't really mean it's validation until I see the effect in the clinic, whether it's the phase 2 trial or phase 3 trial. Sometimes the validations may go up to phase 4. Some drugs I've seen, they're wonderful, they're not toxic, even in phase 3 trials, and when it's marketed released into the market, 5,000 people die in a year, right? So why didn't we predict it? This is where the pharmacogenetics comes in. We are different. Personalized medicine that's the reason personalized medicine is coming, because we couldn't predict these 5,000 people dying through the entire 10 years, 12 years of research. That's where we are at. We need to develop more and more molecular models to be able to figure out what is happening from one person to another person. And this is where the personalized medicine is the only way to solve these translational issues.
I see. Okay, awesome. So I'm just wondering, what according to you will force the industry to adopt in vitro models in their scheme of operations?
Regulatory. So, unfortunately, regulatory still is not mandating these in vitro models as much. Of course, a lot of human in vitro models are being mandated, but they're not yet close to the human in vivo system. So we do use a lot of models, but the thing is, you still need to do a lot of animal research and try to extrapolate your data with PBPK models and PKPD models. Try to come to some kind of a idea, what is my dose going to be, how long is the dose is going to be safe, what is the safe dose, what is the efficacious dose. All these things you're actually using mathematical models only. But these mathematical models could be 30% predictive or 70% predictive. For one compound, maybe it's 70% predictive. For another compound, it may be not even predictive. So this is where these models need to be optimized, and that is where they jump into 3D models that will actually capture more physiology into the system. And I'll give you more information on that, how you can take the 3D model into the more clinically relevant model as an in vitro.
Yeah, so did I catch you right? Did you say that only the regulators can force the industry to adopt? Did you say that regulators?
Yeah, that's unfortunately, yes, that's the way. The reason is because
but on what basis the regulators also would have an authority?
They need data. They need data, right?
It's like a vicious circle, right? So
Right.
who gives the data? Who submits the data to the regulators in terms of encouraging or making certain methodologies or models mandatory in terms of adoption?
So the way we did was back in 1990s through the 2000s, there used to be some pharma organization. Many of the experts from the pharma industries come together. They all know what the issues are. And then what they do is they come up with an idea, say, okay, let's write a white paper with these issues. And they write that white paper, and that actually gets the attention of the FDA. Oh, these are the issues that we are not still looking into for successful drug development. And then the FDA will come, and in fact, I was part of these metabolites in safety testing uh, meetings with FDA quite a lot, with other pharma companies as well. What that meeting came up with is, because if any metabolite that is formed in humans, it is more than 10% of the parent, and if it is an active metabolite, and the other thing is that if that metabolite is lower in concentration in animal models, you have to do separate safety tests with that metabolite. This was the article we wrote about. We said 25% in the metabolite should be, but FDA came and said no, we want anything about 10% should be safety test conducted. So we had a lot of debates between FDA and pharma. And finally, that guidance document was out in 2005 from FDA, how to use metabolites in safety testing. So these are the kind of debates we have participated. Again, the same thing, we had a lot of debates on the in vitro drug-drug interactions. What kind of models we need? What are these predictive models? How do you predict? Because especially Cisapride from J&J, Posicor, which is Mebefredil from Roche, uh, and a few other drugs, terfenadine, they were all pulled out because of the serious fatal drug-drug interactions, people having cardiotoxicity. And these arrhythmias are because of the drug-drug interactions. And to predict that, we had to bring models out there. And in fact, with J&J, I was the guy who actually point person who coordinated the Belgium, San Diego, and Pennsylvania and New Jersey facilities to bring the harmonized protocols together so that we could do it. These harmonized protocols, again, were discussed at length with other pharma companies and make sure the industry follows these norms. Okay, in my own lab, I do my own experiment that is different from Merck experiment. Nobody can figure out what's happening. So we had put some rules around it, and FDA then followed with that. We issued a guidance document. So these are the things that we do between industry, come up with uh, documents or harmonization protocols, harmonization documents, white papers, everything. Try to make sure we have understood the issue very well. We know if we do these things, we can get around this issue faster.
So you mean to say, according to you, there is absolutely no problem with the mindset, or like there is any kind of a resistance in the industry, adopting these
Right.
in vitro or vivo models, essentially like replacing the animals at all.
No, I think the issue when you look at in vitro in vivo correlations, you do need some in vivo data. I'm a big proponent of in vitro models, but at the same time, you don't. All these in vitro models cannot mimic the human physiology uh, much. So you do need the data from in vivo and in vitro combined to generate the models that you can predict the human outcomes. Uh, and we are trying to switch the gears into the 3D models if they can do the same thing without other in vitro models. For example, if I have to do drug metabolism studies, I have about 15, 16 studies that I have to do over 6 months, in vitro studies, to submit to the FDA before I submit to the IND package. So can we get rid of all those 15 studies and can I get all the answers in my 3D model? That would save my time. The other thing is the 3D models are very essential because they're amenable to physiological modeling. They're microfluidic chips, small chips, and your blood flows in 70 kilogram man, where 2 liters blood is flowing uh, in every minute, recirculating in the entire body. But here you're circulating microliters into it, and the amount of drug you need here is less than microgram or milligram maybe. Where if you do animal toxicology studies, you have to make kilograms of drug before you can even do animal toxicology, rat and dog or monkey studies. So these things will change eventually the way we do the drug development. And the good thing for us is that so far industry is saying that animal models are not predictive, but there's some people who also on the other campaign. No, 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 you guys don't know anything. Animals work very well. We have the evidence for it. So the debate will continue for some time until we have these models are updated and as good as uh, animal models in predictive. So yeah, the idea is also that ethics concerns are there. So if these models are as good as predictive as animal models, then you can eliminate animals also.
That's right. Yeah. So, I mean, you've been part of deep academia, and then you've been a stellar participant in the industry research also. So, between in R&D stage and preclinical stage, which is the low-hanging stage to adopt, if at all, these in vitro platforms as part of the operation, according to you?
That's a very difficult question to answer. For the reason is because, as I said, the validation comes in the clinical trials only. Okay, whether I adopt this model in early drug discovery, where you're screening the compounds,
Yes.
or during the lead optimization, or during the regulatory preclinical development, the real validation is clinical data out there. So where do you adopt it? It depends on your convenience, and uh, if you think that this model is applicable at early drug discovery, you could screen. But right now we have almost 50 screens that we go through in early drug discovery in vitro models. The idea is that to consolidate all those models into one, in at least early on, so that you save time and you don't waste a lot of resources and money into it. That's the goal. And the next step is that if these work very well, you can also apply. In fact, Emulate and a couple of other companies are working along with the FDA, and they have some beautiful microfluidic liver organ chip model to predict hepatotoxicity. Of course, that includes the ADME part too, and FDA is actually reviewing the data to see if they can use these models as predictive tools for human toxicity predictions, even in the absence of animal toxicity predictions.
Sure.
Okay.
Yeah, that's good. So now coming to India as a country and the context, where is India industry in adopting these alternatives to animal experimentation, and are they waiting for some kind of push or clue or hint from the rest of the world?
Yeah, I mean, see, India is more of a generic industry in terms of the pharma industry. So basically, very little pharma industry can do it because in generic industry, these are already marketed molecules. Unless they come up with a new indication and they have to validate it, the in vitro models are very rarely used in generic industry. Okay. Whereas if you come to the agrochemical industry or
Yeah,
cosmetic industry,
yeah,
these are dependent on the volume basis because if an agrochemical has to be exported 1 billion tons or one more, I think, you need these studies to be done for every batch to make sure the batch you are exporting doesn't have any contaminants or irritants that can actually cause some problems, and these are regulated in that side. So, cosmetic industry, on the other hand, also is more streamlined now. More and more cosmetics are second generation, third generation cosmetics. So, animals completely are abandoned in that case. So, we are only using skin models. So, whereas for the agrochemical industry, this is the first line. Skin is the first line. If a compound is irritant in the skin model, then they have to test it in the rabbit or some other model to make sure that is not a false po- positive or a false negative. Okay, you have to validate it in a rabbit model or something. So you do need in vivo tests for agrochemicals, but the first line tests are skin model. If these compounds are already on the market, you're coming up with a new formulation, done deal. You can just do the in vitro skin and show that they're not causing irritation or sensitization.
So the data from the agrochemical testing and cosmetic testings on the in vitro, for example, skin models in detail, is accepted by the regulators in India.
Yeah, typically these are regulated by the OECD consortium. India is part of that consortium now. So anything OECD regulates, India also regulates. So OECD committee is also there in India now. and these are reviewed by the OECD committees also periodically, the guidance documents. But we do use guidance documents that are from the Europe primarily. The India, as such, doesn't have any guidance documents on their
Sure.
own. Detailed
Sure.
guidance documents,
Yeah.
except for a few of them, but the real detailed guidance documents are coming from Europe, OECD partners, countries. Yeah. India is one of the OECD partner
Sure.
countries.
Sure.
Okay.
Excellent. This is quite informative, Mani.
This concludes our session, our second session on human surrogate in vitro systems in the testing and determination of safety and efficacy of pharma, cosmetics, and vaccine candidates. Um, this is an ongoing discussion, and we'll explore this topic more in future sessions. Thank you, Dr. Vangala, for the knowledge and insight you have provided us. Thank you also, Dr. Subhadra, for enabling and facilitating this session. To our audience, thank you for listening to this webcast, and we hope you have benefited from it. We hope you can join us again soon for our next session, and we'll be very happy to see you again. Thank you all again, and we wish you a happy new year.